I love it. Got into my brain.
It seems way more of a pain.
No, I don't know. The person that did it yesterday, she was here. Was this sort of a problem? It doesn't seem to want to detect, but what I might do is start talking anyway, because I don't want to run out of time. And I might even introduce you on the board here to a concept called effective population size, which is really important. So how many people in this room know what effective population size is?
Here's the
Couple.
Here's the one.
Okay, I'll let these people work with this. So effective population size is a key concept in population genetics. And another key concept now is that a single genome of an outbred individual in the population can do a very, very good job of sampling that population 1,000 years ago. Okay? Particularly if that population has not been mixing with other populations in that period. And the reason for that is within such an individual's genome, at every site within the genome that's not a sex chromosome, mitochondrial or sex chromosome, you have two copies, one from your mother and one from your father. And if you're outbred, that means you've got 4 grandparents, right? And the basic rule of thumb is that within humans there's 1 recombination per chromosome arm per generation. Okay, so that means that every one of your chromosome arms from your parents has been chopped into 2 pieces. Okay, and then go to your grandparents, it's been chopped into 4 pieces. Okay, great grandparents, 8 pieces. Okay, that's exponential. And pretty soon, you're up to numbers like 50,000, 100,000 pieces. Well, that's actually bigger than most of the effective population sizes of humans in a lineage setting, right? Way bigger often. And so that number is achieved well before 1,000 years ago. So a single outbred individual, don't be looking for big tracts where they've got identical copies because, their great-grandparent, they've married a first cousin or something like that. A single individual can do a superb job of capturing that population. If you were to make it the comparison with your typical autozyme, so your allozymal studies from the past, it's many, many loci with a sample size of 2. As opposed to, for example, a sample size of 10 or 20 individuals with, say, 20 loci. Okay? But it's that huge number of loci in the outbreeding that allows a single genome to capture most of the information you might get about the ancestral population. Yeah.
And when you're talking about allozymes and a sample size of 2, you mean because there are 2 different alleles? Often
with the old allozyme dating, yeah. You'd find anything that would separate on the gel. You didn't know what it was exactly, maybe an amino acid change. Okay, and but these within the genome, within these chunks that have different histories, you get more information than just one version. You often get multiple changes in that region. What's also interesting is then if you look at someone's genome and you sort of say, okay, here's a piece of a chromosome arm, right? This might be chromosome 1, and this is the first region that we can map without ambiguity. So with a lot of this sequencing and mapping, about half of the genome is really usable because you want to map everything unambiguously. Okay, so you don't want to be trying to overlay some of these repetitive elements and things. So they just get edited out. So, the true history might be there was a recombination at some point in the past here, then there was another one here, there was another one here. Okay. If we were, I was going to say God, but if we had God's view of things, as it were, we might say, well, I know there wasn't any recombination in this region, and I can actually build a 2-taxon tree. And the height of the tree is the coalescent age of those 2 copies in that outbred individual. And then you'd say, oh, look, this one here is interesting. It's more recent. And then we go further. Might be another one. It's deeper. What's interesting is most humans, because of our long generation times and moderately good effective population sizes, most human populations have long-term effective population sizes between about 25,000, which might be Khoisan in southern Africa, great deal of diversity. People in this room maybe 15,000 to 20,000. We went through a bottleneck out of Africa, and then we started to expand again. But it was the bottleneck that largely makes us different to most African populations, the bottleneck out of Africa. So what's really amazing, if you look at the genome like this, you can not only estimate effective population size by looking at the coalescent heights and getting a distribution, and the way you do that is that under various types of coalescent theory, the expected distribution under a static long-term effective population size for the coalescent times is an exponential. And the mean, actually basically in time, falls at the effective population size times the generation time. Okay, so if we've got an effective population size, let's say 20,000, and you take the generation time of humans, it's about 30,000 times 2, because we've got 2 alleles, two copies within each. This is the individuals, this is the number of genes, and then multiply that by, say, 30. What's that number? That's quite a big number, isn't it? It's 60,000.
1,080,000 or something.
600 is 1.8.
1.8 million or
1.8 million, isn't it?
Yeah.
Okay, that means that, and this is a big effective population size for living human population. Even Khoisan would be pushing this one probably, maybe 20,000. But this means that you've got diversity that's reaching way back in your genome between all these non-recombined chunks.
So how are those coalescent heights determined? So I kind of understand.
Yeah, oh, very easily. Very easily. Most of the genome is neutral, and it tends to have a pretty even mutation rate.
But you're talking about recombination, and recombination is not random, correct?
Yeah. Now, there's some clever tricks to deal with that, and there's some. We mentioned God here, so putting God aside, modern statisticians have a couple of methods that can deal with the uncertainty of the recombination point, and one of them is called hidden Markov model. It has this hidden state that allows you to say, oh, looks like there's oh gosh, we were here and we suddenly here, oh, there's probably something happened here. And they can do reasonably well at dealing with the uncertainty of where that recombination was precisely. And they're basically, and another one is a Bayesian technique. So they kind of work, and the time back here to a fairly good approximation is simply how many mutations between the two copies are you seeing in that region. And so our genomes, what's also really interesting is this is now, it's a really neat trick you can do, and at the moment most of it's based on 2, but you can extend this to say having 4 copies of the gene, that's 2 individuals. You get more complicated trees, but coalescent theory tells you what they should look like as well under a general mixing population. So what's kind of neat is, okay, this is now, remember, these are big times, this might be 1 million years here. What's going to happen if I want to reconstruct my effective population size 500,000 years ago? Well, to some extent, all I need to do is cut across here and adjust for the fact that I've got this extra piece here and I've missed out some of my loci. And I can make that correction, and I can now figure out what the effective population size of our lineage, or the lineage of the individual with this genome, which would be all of us, because we all share common ancestors 500,000 years ago. I can make a reasonably good estimate of what the effective population size was there. And that has been just some of the dynamite that's coming out from the genome studies that hasn't really sunk in anthropology yet. Now, it's somewhat of an average, meaning that if there was a sudden decline in the population, say the population was 30,000, this is 0, went along like this and 500,000 years ago did this, that would probably smooth out to look something like this. Or actually, sorry, not that deep. So we can't always pick out at the resolution, but we can get the general story.
Can you pick out, say, the Ebonic plague?
In some areas, possibly pretty recent, very, very recent. Not many mutations to do it. About 10,000 years ago, we can certainly pick out Neolithic expansion.
Okay.
And so the resolution of these techniques is better closer to the present. And at about 1 to 2 million years, it starts to get a little sketchy. Not entirely, but more sketchy, more in the sense of, get averages, but we can't sort of distinguish time intervals. And then over that, a little bit. And what's really interesting, I'll show you some of the curves in that, it actually says that for the last million years, our lineage, which we also then start to share with lots of other individuals in Homo like Neanderthals, maybe 600,000 years ago, fluctuated mostly between about 15 and 30,000. Which is really interesting because that's about the effective population size of a chimp subspecies, or an orangutan subspecies, or a gorilla subspecies that hasn't had a recent calamity, or a recent population like the mountain gorilla is a special case. And they tend to occupy areas which are fairly compact. Borneo, Sumatra, part of West Africa. That is going to have major implications as we start to come to some of the other stuff we'll be talking about.
So it looks like I think you've got your slides back.
Yeah, let's get the slides going. But that was probably worth it because that is the power of genomics, that kind of thing. Now, oh dear. Everything's in miniature.
Back the other direction. Just there we go.
Yeah, that's it.
Perfect.
Okay, so here's the first slide.
We didn't even really introduce you, Peter.
Right.
No, I'll do
You're doing a great job.
I'll do an introduction. So some of you may know where Massey University is, but we'll come to that in a second. So my background, my real specialty is methodology for building evolutionary trees and networks. And I'm happy to apply that to all sorts of data. It doesn't really matter to me. You do need statistics to tell you whether or not the fit of the data to the model is good. And if it's good, you're good to go often. And I've been mostly working with applications in the past. Mammals, particularly primates, did some work where we found this group in Japan, Uarconta, which is the big group beyond primates, so primates, flying lemurs and tree shrews, basically. And more recently in the genus Homo, which has both now got fossil data and genetic data and archeological data and biogeographic data and all sorts. So I recently finished a book chapter on this, and conference was in 2015, final version I think went in 2016, and it's sort of a bit confusing. I think they finally published it in March. I don't know what date. I think they've got a 2018 date now, but these were the final proofs, possibly. So I was lucky because I got to do this, and this is the backyard of the place I got to write a lot of the book chapter.
Wow. Oh, wow.
So one of my interests is doing photography, and that was a view off the deck. It's a sort of semi-tropical part of New Zealand, and you've got these clouds, and this is before dawn.
Where's that for people who've been?
This is Coromandel, New Zealand. And then night sky was really nice, just looking straight up. This is just with a standard lens pointing straight up at the Southern Cross. And Alpha Centauri, for those of you that might want to go to another planet. So it was a wonderful place with two major problems. Well, the biggest problem was I had to write a book chapter, and the other one was that the internet was rubbish. No, it was on a cell phone. You might get 1 page per hour or something. So the genus Homo is an interesting genus. It was coined by Linnaeus. And it's not really well defined at all. It's a poorly defined genus. We don't really know when it started, and we don't really have a definition. So what does seem to happen is that after 2 million years within the genus Homo, starting with habilis, brain sizes started to clearly exceed those of apes. So Australopithecines are to a large extent like walking apes, but habilis seem to start a trend. Erectus-like, you'll often see me putting the term like, because I'm a systematist and I don't like the systematics that's been done in the field. A lot of it's very poor. The type specimen of Homo erectus is from Java, okay, and we're talking about specimens from Africa. So whether or not they're the same lineage or whatever, who knows. But they have a broadly similar form, but that's not good enough for systematics. And about 700,000 years ago, first evidence in Africa, particularly fossils like Bodo and maybe Kabwe, which is poorly dated, we start to see a jump. We're getting more than 1,000 cc's, and we're getting sometimes Heidelbergensis-like. Again, Heidelbergensis is maybe not a good name. It's a jawbone from Germany at about 500,000 years. Okay, so how you associate that with skulls from Africa at 700 is a bit sketchy, but it's a grade, as it were. Okay, so something happens in the lineage that is very interesting. That may be the impulse for the origin of the Neanderthal Denisovan lineage. There's evidence of out of Africa, another out of Africa wave of these forms going right through Eurasia. Prior to the Second World War, whenever you found a new fossil that was morphologically distinct, you often gave it a new species or genus. These are interesting. These are the late fossils from Java. Dating is uncertain, but they'll get their new species name Saluensis. Peking man, when first found, even got its own genus. But with the horrors of the Second World War particularly, people like Ernst Mayr, who was an ornithologist, got mixed up in all this. And he decided that we were a chrono-species, which means a single population that evolves through time. The problem was he didn't actually have much data for this. It was pontification. And the proclamation was basically, if it's early, you call it Homo erectus. If it's late, you call it Homo sapiens. And if you don't know what to call it, you make up names like archaic Homo sapiens. Or derived erectus. Make up a name. Great systematics. Thank you, Ernst. And so people would try and do things like this. So we go from this to this.
Is that because of the unclear boundaries based on morphological
I
characteristics?
think it was more due to the horrors of the Second World War, actually, in many ways. It was such a change before and after the Second World War, because, I mean, the Nazis absolutely misused genetics and everything else, right? And, there's incipient racism all over the world, so the idea of talking about a separate lineage can make people feel uncomfortable. But it is biology.
And when you say separate lineage, you're specifically referring to Neanderthal or...
For example, yeah. So if Neanderthals were a completely separate lineage that split off 600,000 years ago and had very little to do with us since then they'd be a separate lineage. And if they kept that distinctness, because they're morphologically very distinct, if they kept that distinctness, then you might well elevate them into, call them a separate species as a systematist. So
is that an existential response to the war, though, and being trying to...
I think it was partly, yeah.
Not see ourselves as just one of many...
Yeah, I think so. Right. I think it was this idea that there were no races, there were no... everything was to be sort of a model of genes diffusing across the world. The multi-regional model, to some degree. Okay, and it was finally the out of Africa model that started to question that, the genetic data for out of Africa, that we had an origin in Africa 200,000 years ago. That means Neanderthals didn't really contribute much to us. And I think the Second World War was one of the big drivers of that change. But you're quite right, the systematics is tough. it is a tough systematic question to tease out what the lineages were. So the idea of Homo as a chrono-species has clearly been falsified recently with perhaps two key finds. First is Floresiensis, which is the hobbit, and maybe only 50 to 100,000 years old. Clearly a distinct lineage. The argument about this, not just distinct, but an early lineage. Some of the features of the wrist hark back to the sort of transition perhaps between Homo and Australopithecus. No one's quite clear. I think if I had to bet money, I'd say it's a derived erectus-like form, but even erectus-like forms are changing. The stuff from Denisovan, some people want to call that Homo erectus. Well, small-brained, different morphology, a lot of arguments about what's what. So there's a hobbit. And the other one is Naledi, which you've probably heard a bit about. Could someone just swing that door, because I'd like to be able to see the clock if possible. Thank you. So we've got until, what time?
1:30.
Oh, we got heaps. Okay. So, so Naledi, big surprise. Cave absolutely chock with material. They probably only excavated like 5% if that.
John Hawkes was here a few months ago.
Excellent, talking about this.
Yes,
yeah.
exactly.
We should have had you guys both at the same time.
Yeah, it could have been interesting. So Naledi looks like a completely separate lineage to Homo sapiens, small brain size, and the age on the stuff in the caves at the moment, 240,000 years, who knows, Naledi might have continued after that. No one at the moment can say exactly where Naledi branched off, when it branched off from us. It may have branched off 500,000 years ago, it may have branched off 1 million years ago, it may have branched off 2 million years ago. The mix of features is quite complex, derived with regard to Homo sapiens and early with regard to Homo sapiens within the genus Homo. And I think John would probably agree with that.
Yeah, he talked about, I don't know if anyone remembers the details, but things in the foot or... Does anyone remember any of that? Like
some of the wrist morphology,
the
joints look oddly early.
Mm-hmm.
Some of the face looks derived.
Mm-hmm.
The brain really small. And just, broad body styles. But body styles don't really tell us about the behavioural differences, and a lot of what was going on within the genus Homo probably had to do with cognition and behaviour and hunting styles, resource exploitation, communication. Again, the reconstructions are very heavily I think approximated by how the hair is done and things like that, whether or not these are that precise, the skin texture and whatnot. And also facial hair. We don't know what facial hair might have been on some of these individuals. But here's a sort of Turkana boy, probably, Naledi, and a loci type specimen. Oops. Heidelbergensis, again, if that was really Heidelbergensis in Europe in the Ice Age, I doubt that it had dark skin. After a while, don't get enough vitamin D, and the hair's all a guess. Neanderthal, same sort of thing. But when you
Isn't Neanderthal and Heidelbergensis, that's not the same?
No, they didn't. Heidelbergensis is a grade, as it were, and there's specimens from Africa and stuff from. No one can agree on what's in and out of Heidelbergensis, and I think that's why we need to build a phylogeny and put individual specimens on the phylogeny. So don't put the cart before the horse. Okay, figure out the phylogeny, then start to figure out species. Do it the other way around, and you'll be arguing forever in circles. So some of the skulls of the smaller brain specimens, habilis, erectus, if you particularly took some of the Denisovan material, Floresiensis and Naledi, we've now got a selection of small-brained specimens, some of which are early and some of which are late. So this idea that brain size increases in the genus Homo depends on which lineage you're looking at. Here's a... Did we ever get a pointer stick?
There's one.
Oh, wonderful, thank you. Oh, interesting. Okay, so...
it's for archaeologists, right? It's rulers.
Yeah, I just want to make sure it didn't have brass on the end, or I might modify your TV. So here's sort of like one idea of, it's not really a phylogeny, it's a sort of a graphical depiction of what might have happened. You've got sort of habilis type grade back here, and you've got sort of erectus type grade, and then this is, the idea is that erectus goes out of, and actually now the date for this is probably even earlier, because the stuff from George is 1.8 million years. So this thing is antecessor in Europe, about that sort of age, 900,000 years, very incomplete specimens. Whether I think the general feeling is probably a dead end from an earlier migration. And Peking man and Java man and Floresiensis may be East Asian experience. Now, this back here, the African specimens have been called Agaster. Okay, so no one can even agree whether or not you should call that Erectus or Agaster. So Erectus really is, it's not even shown here, it's a distinct lineage probably at even 1.5 million years ago from Java. So even this may actually be wrong. We don't know that these lineages weren't separate right back to about here. Okay, just an example of how approximate all this is. Then we seem to come to this Heidelbergensis grade. This is actually too late. The molecular dating now would take that to at least 600,000 years. And we've got Homo sapiens continuing in Africa. I'll be arguing it's just one of many African lineages, partly based on the effect of population size stuff we're reconstructing. And the idea is that all this stuff is guesswork. Naledi, who knows where it comes from. And we've got another out of Africa event and Denisovans and whatnot. Okay, so just a...
I can't read what's on the big thing.
Oh, yeah. So basically you're a robustus stratifications. So in systematics, there's a really important concept that good species, living species, are basically defined as a extant crown plate. And what that means is you build your phylogeny, and hopefully it's fairly tree-like. If it's fairly tree-like, you're not getting more than 10% or 20% gene transfer between lineages. You can identify a clade with a node, and it's an extant clade if there's a living form on both sides of the split. Okay, and in a second, we'll be talking about humans, and we're actually lucky to be in that situation with Homo sapiens, because it turns out we've got very little contribution outside of a common population, a fairly compact population of 20 to 30,000 individuals, and the deep split is still preserved well by the Khoisan people of southern Africa, some of whose genomes show only about 2% of mixing with anybody that's not southern African, which is remarkable. That's basically isolation in southern Africa for, and we'll talk about the period of time, but the best guess is at least 150,000, probably 200, maybe up to 250. So, extant crown plate. Okay, we've got a really nice definition of Homo sapiens. And the reason it works so well is I can reconstruct the ancestral population with confidence. I cannot talk about with any confidence what happened back here. It's all guesswork. But I can tell you with a great deal of confidence as a biologist that that ancestral population for Homo sapiens, if we define it that way, had symbolic thought, had complex language, had religion, and so forth. All things that we share with Khoisan people. And that's really important, that we start on a firm foundation. So, if we define Homo sapiens that way, we can now start to talk about what is Homo sapiens and what's not. Here's a sort of another image of what might have happened approximately, and this is refining all the time, in the last 600,000 years. So here's the Homo heidelbergensis like. I'd like to put like on all of these because it's not a species necessarily. It's not a lineage. It's a grade. Maybe somewhere out here. Guesswork. You know, some people have claimed that into the Khoisan some very deep lineages. I haven't looked at that data myself, and maybe not. Okay. Neanderthal, Denisovans, and quite possibly lots of other things in here that we don't know about at the moment. Very poor record. Denisovan, not even a single skull. Genome, no skull. The stuff from China between 150 and 300,000 years has multiple skulls, but they're quite diverse. It's not clear you can put them into one box. Even in Europe, 250,000 years ago, does anyone know what the Petrolona skull is? Beautiful skull from Greece, 250,000 years old. Doesn't look like a direct ancestor to Neanderthal. There are clearly Neanderthal-like forms walking around based on some specimens that have been redated from Italy at nearly 200,000 years. And this thing doesn't look anything like them. So another lineage, right? So a complicated picture. This part of the story is really interesting. Here's Homo sapiens. I define it as the last common ancestral population before that deep split within living humans. And all descendants. And I'll allow up to 10 to 15% of other genes from other sources and still call it Homo sapiens. That's the type of compromise you have to make as a systematist. When you get beyond that, say 20 to 30 to 40% coming from one parent and the rest from another parental lineage, then you're talking about a hybrid species. But we're not like that. We're not a hybrid species. We're actually a pretty well-defined, compact, single lineage species. And within living humans, the structure that's emerging is, and this structure is what the world might have looked like, for example, 50,000 years ago. Okay? And it's been, you know, there's been mixing over time, but trying to capture some of the deep early splits. Khoisan, Southern Africa, deep split, extant. What's not shown on here is that there are click languages. So the Khoisan people often make click sounds when they talk. It's a consonant sound, but it's created as a click sort of noise. The only other place you really find that, some of the Bantu groups that have come into Southern Africa pick up a few of these, but the only other place you really find it is a couple of isolated populations of hunter-gatherers in Tanzania, the Hadza and the Sandawe. And they've been of great interest from the earliest out-of-Africa days, and complete mitochondrial sequencing has suggested that they share some deep lineages with Khoisan. What's interesting is a few years ago, and some work done at Harvard University of Chicago, they sequenced Hadza and Sandawe genomes, and they tried to subtract the Bantu signal, the West African signal, agricultural signal that swept through there in the last 5,000 years. And when they did that, their claim was that they got a deep lineage from Tanzania. And actually maybe two. Hadza first, closer to Khoisan, and a deeper one for the Sandawe. That work is still needing refinement. The other surprise was that the effective population size in those Tanzanian lineages may have been bigger even than the Khoisan. So effective population size is not really well shown on this, but the Khoisan are basically the most diverse living people. And then there's a clear branch to pygmy-like people in Central Africa. Again, there's some admixture with West Africans, but when that's subtracted, you get an even cleaner signal for the split. Then there's West Africans, and they may not be even a single lineage. We'll be talking about two genomes later, one Mandinka and one Yoruba, that don't. They join very deeply, if at all, before they meet the common ancestor. And East Africans, Dinka are a deep lineage. out of Africa now, looking from the archaeological evidence, perhaps like going to Arabia first, 100,000 years ago, and then out, and then the rest. And, you know, some contribution from Neanderthals and Denisovans. The average across people that can trace ancestry to Eurasia, 2 to 2.5%. Chopped up into small chunks now, and we'll talk about that a bit later.
Can I ask one question before you go forward? When you say the Khoisan are the most diverse, now is that because of a large populations, effective population size at one point, just a deeper lineage so there was more time for mutations, or just because of the bottleneck of other populations moving away?
All lineages are equally deep. We've all had the same time for mutations. Okay?
Okay, so it would only be the other
it's
two.
Basically the average effective population size between the Khoisan and back here is larger on average than to any other lineage.
And essentially the bottleneck and effective population size are kind of related. They're kind of
yeah, if you go through too many bottlenecks, like out of Africa, you lose diversity,
right.
You lose effective population size over the long term. So some of my work, I was the first person to figure out that the Denisovans actually have a few percent coming from something much earlier. And I'll just call it the rectus-like. It's just prior to this split. Some of the more sophisticated methods now are saying that they're dating that material at 3 million years. I'd be surprised at that. There'll be Australopithecine. I think they've got some problems with their calibration, but I could certainly believe it was erectus-like. You know. Erectus-like forms may well have been walking through Asia 1.8 million years ago. So the other split I'll be showing you some data for is there's now some evidence that somewhere in here another lineage was distinct in Africa, and in West Africa, particularly Mandinka, picked up maybe 6% of their genes from that lineage. And Yoruba maybe a little bit, couple of percent, but we don't see that signal into these other African populations. We'll be talking about that further later. So yes, there is little signals coming in from other lineages, but this is the biggest single one. Some Australians and Papuans may have up to 5% preserved of that. So effective population size, I spoke about that. A little bit of a boost before our species originated. Here's some of these reconstructions. Here's this. It's where the lines diverge. This is a pairwise method. As I mentioned, it's pairwise. Some of the more sophisticated ones now will build the trees from more genomes. More challenging computationally, but can be done. These are the pairwise ones. So this is from 2014, and all the lines really should be very similar back here. And here's your Denisovan and your Neanderthal, and they sort of start to look a bit different perhaps around here. And then here we start to see some of the Homo sapiens lineages start to diverge. Here's the same. And declining effective population size through time, but still enough to keep the early diversity because they're higher in this period. And this might be around 200,000 years ago, 250. Some people have even suggested 300, which I find a little bit too old. The idea of modern humans wandering around Africa for 300,000 years is pretty remarkable given the archaeological record. And there's that dip that I mentioned, and then we'll climb back.
What does it mean when the Denisovans and the Neanderthals drop to zero there?
Well, they probably disappear. It seems that their lineage, out of Africa, was either here or here, depending on how you interpret this. This could have been the mixing with earlier lineages, possibly. But they drop, they go stable for a while, and then they just before those specimens were taken, those lineages drop. But we now know that situation for Neanderthals more complicated, because that is the Neanderthal that is in the same cave as Denisovan. This is this cave in Siberia, where Kazakhstan and Mongolia meet on a map. Go north a few hundred kilometers, and that's where the cave is. It's basically above East China. You go south and you hit East China. And so what we do know now is that from multiple genomes of Neanderthals, is that this is just like an outlying population. And its split time from the Western Neanderthals might be about 200,000 years. 150, 200,000 years. So we're looking at, for Neanderthals, a fairly compact, distinct form. Another group that might well be defined as an almost extant clade. When I say almost extant, we've got their genomes, but we don't have their form. And until we can link genomes to phenotype, we don't really know what their common properties were. And so Neanderthals seem to have multiple lineages that split off, occupied different regions of Eurasia, and eventually disappeared. But a complex structure, a bit like the complex structure we see deep in Africa, for example, amongst Homo sapiens. Now, that's the situation 50,000 years ago. Go to Africa now, and it's dominated in sub-Saharan Africa by West African genes because of the agricultural revolution. And most of northern Africa is dominated by Eurasian genes because of a back migration that started about 40,000 years ago. So Egyptians are basically Eurasian-derived, mostly. Okay? So Africa is a complicated place. That effective population size is about the same as 8 subspecies. And 8 subspecies, and 2 ways of measuring effective population size are more ancient, more recent sometimes. This is suggesting bottlenecks more recently. So if we take some of the African populations, they've neither experienced recent strong bottlenecks nor more ancient ones. But we overlap with Western lowland gorillas and Sumatran orangutans and central chimpanzees have got a bigger effective population size than us, okay, but in both regards. The orangutans are out here, they're a little smaller in one regard, but they're the same in terms of the more recent history and so forth. So we're not atypical. Some of these are Neanderthals, but remember these are all Neanderthals, so suggesting a diverse, highly fragmented structure to their population that went through bottlenecks probably. And eastern lowland gorillas, and where's mountain? I don't know where the mountain gorillas are. I think they're considered eastern lowland. And so Denisovans and Neanderthals got fairly small effective population sizes. But the common ancestral population size of all the Neanderthals, you'd have to go back 200,000 years perhaps to when they originated. It might have been more reasonable. It might have been 5,000. What's interesting though is that even though the population size was smaller, Neanderthals do seem to be evolving. Neanderthals show, that lineage shows now some evidence of symbolic thought. Okay, so, which the ancestors 600,000 years ago may not have. And Africa has many regions that can support populations of that kind of size. So Africa's a very big place. And it's possible that in the right environment, you could have fit our ancestors into an area like the Blue Square. So when we say Homo sapiens came from southern Africa, what we mean is that it came from somewhere in southern Africa, and as yet, we can't be sure where. This is, I think that's Tanzania. Maybe some evidence for high diversity there. Down here, Central Africa, Where it all started, bit of a mystery still. We may actually need ancient DNA if we can ever get it. Africa's not such a good environment for ancient DNA. Ancient DNA. DNA does not like hot and it does not like wet. And Africa, a lot of areas in Africa don't do well on both regards. There's now a little bit of evidence coming out of samples through this region that are 5,000 years old. And there was one that I think from this region that turns out to be, as it were, Khoisan, and it's only about 5,000 years old, but the mixing of non-Khoisan genes is even smaller than 2%. So, given that we've got a definition of Homo sapiens, which is really, really important, you won't believe that there's all sorts of definitions of Homo sapiens out there. Basically, if it's not a Neanderthal and it's derived more recently in Africa, some people call it Homo sapiens. Okay? Well, I don't have any idea as a biologist what the properties of that lineage would have been. That's why I think it's a very bad term to use. Pre-sapiens is okay. If you want to call pre-sapiens with regard to Neanderthals, that's fine. It's pre and from. Or Homo sapiens like, that's less defined. So we can diagnose, but not define, a Homo sapiens based upon some features of the skull. These are features that all modern humans seem to have, living humans seem to have, and some of the well-associated fossil forms, like Cro-Magnon man. The definition is phylogenetic. The diagnosis for morphology is trying to find what's called a really good synapomorphic, a feature that's derived really, hopefully, just in that ancestral population, and then fixed in all the descendants. And there's one particular feature that seems to almost meet that criteria. Getting a great synapomorphy is not easy. You may never get it morphologically. But it's the inverted T-shaped chin. Okay. So if you feel your chin, it's not a single structure. First of all, there's a piece that juts forward. Neanderthals don't have that at all. Then there's a keel. If you actually feel the midline, there's a keel. There's a ridge that runs down the middle. I see that most of you have already done this, so you don't need to verify that you're a Homo sapiens. And then at the bottom, it sort of flares. This is a square jaw. Okay, you remember the Superman comics? Superman's got a very square jaw. Superman probably was a Homo sapiens. And at the end of that, you often get two little bumps, very sharp points, and you've also got some thickening underneath as well. That feature is clear almost from birth, even in embryos sometimes. Sorry, fetuses. And this is very early stage where you can see the sort of start of the development. Sorry, this one here, the cleft. Yeah. This is a 1-year-old, I think. That's a 2-year-old, that's a 5-year-old, that's an adult. Not aware of any living human that doesn't show at least 3 of those 4 features of that inverted T, and most individuals show all the features of the inverted T. First appearance in the fossil record, remarkably late. Kafsa, Israel, only 2 of the specimens. Other specimens in the cave, no, don't have it. Classes Rhythmouth, a couple of specimens. Other specimens in the cave, no. Both about 90,000 years old, both at the extremes of Africa, as it were. And it's present in this early European that maybe had a Neanderthal great-grandparent. So it may be dominant characteristics. Okay, so you can throw in some other genes there and it still gets expressed. The second feature is a bit more problematic. There is some evidence that some of the late lineages that diverged before Homo sapiens may have had this. The bipartite brow is not the fact that there's a brow over each eye separated by a glabella that's not sort of linked to them. It's actually the fact that particularly in adult males, there's a cleft that divides the brow into two pieces. So if you actually feel your brow, you'll actually feel that depression. There's this structure here on the side, and there's this piece up here, and there's this cleft that runs obliquely across. That develops in most males, and it tends to get stronger as they get older. It develops in some females, and that development in both males and females, if they are going to develop it, tends to start in adolescence. The problem is that if you don't develop it in your Homo sapiens, you have what's effectively a primitive condition. Like a young Neanderthal would look the same. So if you have got this, you're close to Homo sapiens. If you don't show it, you show the primitive condition. If you show one of the other brow types, Neanderthals have what are called aviator goggles. They have this thick brow, it's uniform, right across, nice arc, no dip in the middle. Okay, got one of those, you're not a Homo sapiens.
Is this, though, it's arising during adolescence, is this like hard genetically programmed? Is this
all
hormonally kind of development?
Yeah, no, it seems to be all populations checked so far have it, of males particularly, yeah.
But the ones that don't, or the females that have it versus the ones that don't, it seems like there's either a genetic variation there or there's an environmental difference.
Yeah, so what we say in genetics is the penetrance is not 100%.
Okay, Natalie, I'm wondering, just thinking about looking at that picture, obviously that bipartite situation is a product of that suborbital notch of Freeman at that location. Do non-human species have a suborbital notch of Freeman at that location?
Yeah, they don't seem to show the full depth. Have I missed? We've got to one.
No, we have some people have to go.
Our
Okay.
graduate students have a class they have to be at.
Gotcha. Okay, so
Those were the developmentally, that was a product of anatomy.
I think if you look at the Neanderthals, there's just nothing going on
there's
like that.
nothing there.
Yeah.
I didn't think of.
Yeah. I think the foramen might be actually underneath slightly. I'll have to check that. So even recently, if you look at Stringer trying to explain the origins of Homo sapiens, he's a very popular writer and researcher on this. He will present a figure like this, which looks a bit like a chrono-species through time. Right? So these are some of the African specimens, and he's got them lined up effectively in form and time to some extent. This one is actually sometimes regarded as the first skull that shows a shape close to Homo sapiens. It's now dated at 195,000 years, but you can see it's very fragmentary, and it's only shape. Doesn't mean it's got a bipartite brow, doesn't mean it's got a mandible associated with it. This one here, I've analysed data from it, and we'll talk about that. That shape comes out mixed up right with even Khoisan, for example, very, very clearly a Homo sapiens shape. This one doesn't. This is actually from the same period in Israel. Does not appear as a typical living Homo sapiens. So those features also argue that we came from a restricted population, because we don't find this T-shaped chin all over Africa, for example, at 200,000 years. So our species doesn't really show up in the fossil record, if at all, until around 90,000 years, and maybe even later. There's lots of stuff also in Africa that's less than 90,000 years, some of it down to perhaps 10,000 years, that does not have the inverted T-shaped chin. So Africa was a complicated place even up until relatively recently. And the fact that we've got that...
Sorry, can I ask a really naive question?
Yeah.
But then why does that suggest that that's a good diagnostic criteria, right?
Because all living humans have it.
But, I mean, that I totally understand. But if it's, it seems like there must be something else that we could use that is a little more consistent, right? Or is that as close
It's
as
highly
we can
consistent. As far as we know, all living humans have it.
Sure. In modern times. But if the spread of it, if the spread of the appearance is that large... It just seems like...
No, the argument is that these other forms are not Homo sapiens, right?
Right.
They, as defined as the descendants of that last common ancestral population. And that includes some specimens from South Africa, a couple of skulls unrelated to each other in terms of finds. One is fishhook, I think the other one is washcott, dated maybe 20 to 40,000 years, mandibles, no inverted T on the mandible. The shape of the skulls are fairly complete. Shape is really close to us. Okay. So what's happening, for example, at Classy's river mouth at 90,000 years ago, we don't know. Does that mean that there's changing populations? There's a population that's pre-sapiens and then sapiens is moving in? Or is it a mixture, hybrid population? We don't know. Okay. And the same thing with CAFSA. Don't know what's going on there.
But so that's the best diagnostic criteria that we have. That's the one thing that we can
The
prove
chin,
that is
yeah,
totally...
yeah. And so if someone says to you, I've got a fossil that's Homo sapiens, and you accept my definition of Homo sapiens because I think it's the right definition biologically, and to be precise about properties, then if it hasn't got that, you must query it. And the stuff coming out of China at over 100,000 years so far doesn't have it. There is a Chinese specimen at 60,000 years, and we've also got some DNA out of it, which is clearly a branch of the modern lineage. It does have it. The stuff at 100 plus so far doesn't have it.
So I think maybe one of the things that I'm also trying to get my head around, and Stacey, because we're not, paleoanthropologists.
Oh, no.
But it's like trying to be like,
it's
it seems like this weird, very kind of like small thing to be like, we're going to define this whole tree.
No, we're not defining it. I said that very clearly. We're not defining it, we're diagnosing it. It's the best single morphological diagnostic character we think we've got. And it's standard cladistics. This is standard across the whole of all living things, bacteria onwards. And the idea is just gradually creeping into anthropology. Yeah.
Is there a theory on if we use that diagnostic as reflective?
Okay, and first thing I'll say is that I don't know that it's absolutely diagnostic of Homo sapiens. And there's some caveats in the book chapter. First of all, the guys that have done this so far have not checked a lot of Khoisan skulls. That would be the first place I'd start to be checking carefully. If they actually, some of them miss it, that means that Classy's rivermouth population may in fact have been Khoisan related. Okay, so, but certainly from West Africans onwards, in that part of the tree, all specimens seem to show it. Or at least the start of it and pieces of it. So, sorry, your question?
I guess the basic question, because I'll get lost in the weeds otherwise, is how does your phylogenomical definition of speciation relate to like other definitions of speciation, like capability to produce?
We'll talk about that later. Yeah, we're coming to that.
Yeah, because the first half, we set up this kind of genomic.
Oh no, that's absolutely important though. That's a precise phylogenetic definition. Okay, so generally in biology now, the phylogenetic definition tends to have primacy. The other definitions, if they match up, nice. Phylogeny has primacy. Confident in the phylogeny, you can start to mark out clades. You hopefully mark them out so they coincide fairly well with other characteristics, phenotypes, and etc., etc. But extant clades have a special property. We know what we're talking about when we talk about the ancestors and all descendants. Anything else, we're guessing. So, some analysis that I did in the book chapter, I've done it in other places, but the book chapter is a good summary. We're going to build a wireframe in 3D space, and the wireframe is going to follow, this is the coronal suture. The sagittal suture runs from here and back to the inion, inion down here somewhere, and we're also going to follow the lambdoid suture across the back, and we're going to follow the top of the brow, the border of the brow front, and it's going to be a point there and a point there and a landmark there. Okay, so it's going to give you a wireframe of the top of the skull, shape of the skull. And having done that for a good selection of fossil Homo skulls, and actually a couple of hundred living humans from populations as diverse as West Africans, Australians, Australian Aboriginals, Asians, Eskimos, Europeans, all of those populations having at least 10 individuals, sometimes more. You can start to then compare distances between all these skulls, and it's called a Procrustes distance. Only once done shape analysis. This is a simple shape comparison. So you scale them to be the same size, you center those wireframes at the same point, and then you do a rotation to minimize the Euclidean distance between all points. You sum up the distance between all the points, you get a distance. Okay? And then one of the 3 or 4 main branches of phylogenetics starts with a distance matrix and builds an evolutionary tree. It's not phonetics, it's phylogenetics. These distances can be considered a maximum likelihood pairwise estimate. And the model that they seem to correspond to, if you want one, and it's a little bit of a funny model, is that all the points independently oscillate with many, many gene properties. The model is probably wrong, but you can still analyze the data, see what we get. So these trees are general linear models. We know lots and lots and lots about their statistical properties. So it's tree-like. That's the first thing. The tree that we get is tree-like. A tree is a reasonable model as a first approximation. Doesn't mean there isn't some reticulation, but the tree captures a lot of what's going on. Now, here's an interesting example of the diversity in Africa. I haven't got Boschkop or Fishhook in this data set, but I do have Iwe-Alaru in this data set. I didn't collect the data, but the data does have this. I'm trying to add some of the other ones in. This is from Iwe-Alaru in Nigeria. And so if I press this, this should actually change, right?
Yes.
Great. So, 14,000 years old, multiple dates. The carbon-14 date is probably the best. They've got some, I think, uranium dates as well. Is it demoluminescent? But they're all between 10 and about 18,000 years, and I think the carbon might be the best from charcoal next to it. Crude burial, very crude burial, no grave goods. Not a Homo sapiens skull. Okay. Mandible is a bit damaged, but no suggestion of an inverted T-shaped skull. So, let's look at the tree. So here's the tree. Back here we've got some Agasta skulls. We've got Heidelbergensis-like skulls here. I've constrained the Neanderthals to fall in one group here. Then from here onwards, we're talking about African lineages basically. And right up the top here is a Khoisan. Okay, there's actually 2 Khoisans. Khoisan 1 and Khoisan 2. When I add in all the other modern skulls, 67% of them would appear branching below that point. 2/3. A few modern skulls are outliers. Some of the really outlying ones are there's an Aboriginal male skull and, curiously, a female European skull. Okay, but modern right in here, there's Calf-9, right in there. This is constraining Cro-Magnon to be a single group and constraining the pre-Cro-Magnon cultures of Europe to be a single group. What's interesting is the suspicion is these are holding a higher frequency of Neanderthal genes. The estimate now is for these ones probably around 6% average, or maybe even more. This OSA2, which may have that Neanderthal great-grandparent, these ones may be around 4%. And when you get hybrids in a tree, they're sometimes sucked towards the other parent. And what this is showing is that with considerable confidence, these earlier ones branch deeper than the later Cro-Magnons. which would tend to agree with the estimates of how much Neanderthal they've got in them. So when people have talked about discrete features of these early European skulls that sometimes echo Neanderthals, and there are a few, and may well be right, this is actually based on overall shape, and it's suggesting that these early Europeans, because of Neanderthal influence, are getting sucked back down the tree. What about the rest of the tree? Well, all of this is Africa from probably 600,000 years ago. Kabwe is poorly dated, anywhere from 200 to 1 million years old. This would suggest it's actually pretty early. There's Ewo-Elaru, way outside. This is a very clearly defined point here, way outside of anything near modern. And you could sort of approximately date that if this sort of Khoisan thing here, say modernity somewhere in here, say 200,000 years, and then 600,000 years, you might say that's, and there's some rate changes going on, you might say that's 400,000 years ago, maybe a little older. What's interesting is there's another skull in here that's called Homo heidelbergensis, like that's Saldanha. It's a skull cap from southern Africa, also from Springbok Flat, Edelfontein. The names keep changing depending on the politics. And it doesn't come out anywhere near Kabwe. And in fact, it may be one of the first skulls we've got on that lineage going towards us in Africa. And these skulls here, Uhud, Jebel Uhud, they've been grouped together here. They otherwise appear like here and here. They've recently been redated to around 325,000 years old. The oldest, I won't say Homo sapiens, they want to call them Homo sapiens, they're not Homo sapiens. But an early example of the lineage. And whether or not they're ancestral or a side branch is unclear. Probably a side branch, I'd say, but who knows. And then we've got all sorts of other things, and a lot of these appeared in Springer's Great Chain of Being, but we're seeing deep branches. What's interesting, in some analyses, different tree building methods slightly, Saldanha groups with Ewo-Elaru, groups with LH18. Maybe another clade spread highly diverse in biogeography and time across Africa. Don't know. Not strong evidence yet. But we need to start looking at trees and not simply assuming there's an ancestor-descendant relationship. Ewo-Elaru certainly looks like a deep lineage. So what are whole genomes telling us about the situation? So one of the first really great data sets is from Waddell. They've got the Denisovan, they've got an early Neanderthal genome, and they have got a lot of diverse, at that time, 5 very diverse living Homo sapiens populations. Okay, so Denisovan comes out closer to Neanderthal, but only slightly in some degree. And the only part of the tree that doesn't really resolve is French hand Papuan. We can talk about that. It actually looks like there's a bit of interesting stuff going on there. So I'm going to show you the analyses I did of this, different methods to Reich et al. There's a tree. This is the only bit that doesn't resolve clearly. But this is what's called a network. And it's reconstructed with a technique called neighbor net. Now, building networks is much harder than building trees. The number of networks is vastly bigger. Number of trees is already huge. It's a double exponential. By the time you've got 20 tips, I think, was it 30 tips? 20 tips, I think, you might be talking about more possible trees, binary trees even, than all the elementary particles in the universe. Okay, it's a big number. But we're getting pretty good at it, as long as the data is somewhat tree-like, figuring out what the best tree probably is. But this technique seems to do a pretty good job of pulling out a network. So it's tree-like. I've just chopped off all the external branches, but we've got the outgroup, the chimp. We've got this branch going to the Denisovan and the Neanderthal. That's that little branch in there. Then we've got the branch going to the living humans. We've got the sand coming off first, then the Yoruba. And then this one actually says it's the French first, and then the Han and Papuan. Now, this actually also says there's a signal between the Han and Papuan. Historically now, the data that's coming out is saying, yeah, that was the original split. So coming out of Arabia, we've got West Eurasia first, then a split in East Eurasia with people going into Australia. And then since that time, we've had some migration back and forth to a certain degree across Eurasia. Think Mongols, think, actually it's really interesting. Native Americans actually trace a lot of their ancestry back to West Eurasian. They were a mix even 30,000 years ago. Okay. This is a data set 2012. We've now got, using the Denisovan as our group, we've got the San, a Pygmy, 2 West Africans that separate deep, a Dinka, out of Africa, West Eurasia, Papuan. That big split there is the Denisovan genes coming into the Papuan. And we've also got Native Americans splitting off from East Asians. Okay. Through here, we're also seeing that the Native American actually splits somewhat with West Eurasians. That's been verified now with some fossil material from 30,000 years ago. This split here doesn't actually always appear. These three sometimes may congeal. More often, sometimes Mandinka will come as quite a separate lineage in here. Different methods of analysis. So that's the general picture, and the Mabuti share some with West Africa. That's everything we can fit on a planar graph, and I've also trimmed it so that all the edges upon resampling have at least 80% support. These are these numbers here. Sorry, 75% support. This one is a little bit weaker. And networks, it's much harder to get support because there's many, many, many more competing edges than in the tree. Let's look at the residuals. Let's go into the residuals because these are things that we can't put on the plane. And here's your QQ plot. Yeah, it's looking pretty non-linear out here. It's actually too many genes shared between Mandinka and the outgroup, the Denisovan, and too many genes shared between Mabuti and Dinka. That Mabuti-Dinka thing is actually historically approximately correct. Dinka take Mabuti brides sometimes, or Pygmy brides. Take them out, and we're going to take them out by creating their special splits just for those interchanges, and everything starts to look pretty normal, and doing sums of squares, chi-square type statistics, big improvement in likelihood. These are these two events. So, here's the diagram. The first one we can put, and it's basically a bridge. We can't draw it in the plane competing with these other edges, but we can go into the residuals and create a bridge. And it says that genes were shared between those two lineages. And that's the other bridge between Mandinka and Denisovan. This one's really kind of interesting because this is pre-sapiens. It's in the pre-sapiens direction. We don't see those events. We see the first one on chromosome X, but not the latter. Okay, and we don't see this with Denisovan or Neanderthal. So X chromosome does not like these mixings. Here's the X chromosomes trimmed down. We've got that one, but there's no signal between Papuan and Denisovan, okay, on the X chromosome. So what's a species? This is the question. So phylogenetic species, reasonably easy to define. Pick a group that looks to have some common phenotype, genotype, something, and put them together. That is a single clade, single lineage on a tree or network. Biological species complex. Populations interbreed readily, but not so much with other groups. Okay, well, bit of a problem. You can take all sorts of things that we consider good species in the wild and you put them together in the zoo and they mix up. So we certainly do see evidence that when populations of the genus Homo encounter each other, there might be evidence initially of some hybrids. But there's another definition, it's a co-adapted genome complex. That selection has chosen a gene here, a gene here, a gene here, a gene here, and a gene here. Okay? It's a bit like trying to mix a Ford, the parts off a Ford and a GM, right? Both got steering wheels, okay? Would you swap your steering wheel? Okay? You're going to have trouble. So the co-adapted genome complex means you start swapping these around and you drop your fitness. And you may drop it so much that natural selection will weed you out. So this is this hypothesis of a deep lineage mixing into West Africa. So back in 2011, these guys suggested some deep, but they also suggested deep lineages for San, and they also suggested for Pygmy. Okay, so it wasn't too specific. Archaeology says the transition out of the Middle Stone Age is remarkably late in West Africa. And we've got that deeply divergent skull from Nigeria. Turns out that just recently, the deepest Y chromosomes in living humans come from the Embo people who are actually from near Nigeria. Almost twice as deep as Khoisan. And we've got this autosomal signal. We're seeing something that's pre-sapiens coming into Mandinka in particular. The asteroids are my work, the other ones. Okay, and in my work, I can show that the Y chromosome and Ewo-Ellery skull seem to branch at roughly the same point. So, just to finalize, so it's another piece of evidence. It's looking quite good that West Africa was another mixing between a discrete lineage and Homo sapiens. And if you're going to accept a phylogenetic definition of Homo sapiens, and you might not like it for some reasons, right? It doesn't tell us about what was before, okay? But it's the only definition that we really can put on a firm basis, because we can talk about properties of Homo sapiens. I don't know what properties a Neanderthal had. Okay, so first of all, extant crown-based definition. This is really what came out from the mitochondrial work and has been verified over and over again with genetic data. This term archaic is unusual for systematics. It kind of means primitive and ancestral, but how are Neanderthals primitive and ancestral? They lived contemporary with our ancestors. Contemporary things are neither primitive nor ancestral. That's the point. And human, I think, is best associated with Homo sapiens alone or not at all. Okay? And we could call Neanderthals near human. I don't think we should call them human. And this is going to muddy, when we do this, it muddies the traits that we share in common as living humans. And it disregards or denigrates what are called Neanderthal traits. We don't know what they are. We've got the genome, but we don't really know how Neanderthals functioned. There was one thing I'll just jump back to very quickly. Okay, which part of the brain evolved the most? This is pretty much the end of the talk, so we've got a second to jump back. Okay, which part of the brain do you think evolved the most since we split with Neanderthals? Are we talking about, of the major lobes, frontal lobe, parietal lobe, occipital lobe, or temporal lobe? All big lobes of the brain. Okay, anyone who wants to put up their hand for frontal? Okay, who wants to put up their hand for parietal? Who wants to put up their hand for occipital? Okay, and who wants to, two. And who wants to put up their hand? You must be putting up your hand for temporal. Okay, so actually, nobody got it right. It's almost definitely and clearly the parietal. Neanderthal skulls on that tree don't actually change a lot from erectus. They grow bigger, they grow really big actually, bigger than living humans, maybe almost about the same size as Cro-Magnons. They grow kind of like this, okay? The base of the skull, the torsion angles that you can measure across the foramen magnum, where the spine comes out, is an angle you can measure. It's flatter. Modern humans, that starts to get like this. Modern human skulls sort of, okay? Parietals, getting longer and bulging at the top. Our skull is wider up here than it is down here. Okay, Neanderthal skull from the back looks rounded, and erectus type skulls look almost triangular. Parietals have expanded dramatically in our lineage. What's interesting is that there is some evidence, in fact, that parietals were also expanding in Neanderthals. Okay, and we do know that from imaging studies, parietal frontal, you can actually measure a person's IQ to some degree by having them do tests, and you can look at the brain pattern between frontal and parietal, and with a correlation of about 0.4, which I think will grow as I get machine learning onto this, you can predict the person's IQ from how their brain is operating. And it's a parietal frontal talk, back and forth. There's a lot of suspicion that things like episodic memory and consciousness may have critical components in the parietal. Okay. And it's a relatively late evolving region, at least expansion in us. So I'll just conclude very, very quickly. We've only got one more slide, I think, if that. So out of Africa today, well, it's the basic story. I think it's probably in this range. Some people are pushing much earlier for the split with Khoisan. Somewhere in the south, possibly quite a limited area. So hopping possibly through Africa, not a single event. Possibly encountering other groups. So it may not have just been the environment that was keeping us out of these other areas. It may well have been relatives, related lineages competing with us. Some interbreeding and then declining frequency. So we know in Europe, some great studies came out. Look at Cro-Magnon, 6% Neanderthal. Trace it through time. Hunter-gatherers right before the Neolithic, down to a 2 to 3%. Neolithic down to around 2%. Okay, so natural selection is weeding out Neanderthal through time. And lots of species competition, and that's the end. So the story that's emerging is a much more complicated one than we assumed. So I better take some questions quickly because I've run over slightly.
See if Chris.
Yeah.
Oh, okay.
He emailed it? Okay.
Well, I have a quick question to sneak in. I feel like there's a dance that you're doing, intentionally or unintentionally, between phenotype and genotype.
Yep.
Like there are some connections made between the genotype, for instance, say parietal expansion, what that means by thinking about consciousness and symbolic thought, and things like that. In the literature in general, how much dancing do people do? Like, can you actually look at the regions of the genome that vary and correlate it to
Parietal development.
digestive expansion or limitation or...
Yeah, well, that's really just starting. So,
I mean, it's you know, sketchy.
Yeah, absolutely. Ask the same question in 20 years' time,
Sure.
and assuming everything's going well in the world economy, generally we'll have much more precise answers. We do know that there are some genes that are involved in parietal development in Homo sapiens, okay, that are on the X chromosome, and those forms were not replaced by Neanderthal coming in. So in other words, they're really poor in Neanderthal genes. There is one gene that may be related to parietal that seems to increase in frequency in some Europeans. which is an interesting one. I think that needs a lot more look at. Generally, the only things that increase in the Neanderthal sort of infusion are some immune system genes. They're like playing cards, you know, different card, yeah, great. Diseases in Europe, fantastic. Very modular, very discrete. And possibly something like hemoglobin for high altitude.
Sure.
Yeah. So tends to be a suggestion that there was some co-adaptation, maybe. And another hypothesis is that the lower effective population size of the Neanderthals created more deleterious forms that were weeded out. Yes and no. You know, Neanderthals were still evolving. They weren't their ancestors. And you know, a little bit of deleterious. If there were a few really good things to switch in there, you probably would. The car analogy, right? Okay, I've got an AC Delco part in a piece that's been sourced from some country overseas that's poorly made. Well, maybe not poorly made, don't know the quality, but it's probably still worked pretty well.
Yeah.
Yeah. So, yeah, 20 years' time, we'll have a much better idea of what how these things might translate.
I was just thinking, like, prior to our modern understanding of genomics, right, everything was about phenotype. Everything was about just what you saw, what it looked like. And so it's interesting to try to piece that into you know, what you can do now, which is so cool and so much more details.
Yep.
You know, but people are still interested in.
I'm just.
You show all those recreations of all of all the hominids, right?
Yeah, I mean, I'm just learning about some of these brain imaging studies, which I think will become relevant too. It's like stunning that you can actually kind of measure someone's IQ to some degree on certain types of tests by that. And I think that, again, with more data and machine learning techniques, particularly to pick up particular patterns that aren't obvious, we may well start to get up to much higher correlations at some point in the future.
So Chris just emailed with a question. He says, terrific talk, he really enjoyed it and learned a lot. So what he wants to know about is your reconstructions of the effective population size.
Yep.
What's not clear to me is when the effective population size of 25 to 30,000 existed.
Sure.
Is this in reference to the common ancestor of all living humans, aka the crown of the human clade?
Yeah, okay, so let's jump back. It's not my work, it's taken from a Nature paper. I explained the principle basically here, but there it is. Okay, so this is the first paper with the resequenced Neanderthal genome. Different Neanderthal, the one from Altai, much better quality. Same cave at Denisovan. That cave preserves DNA really well. Okay, so we need to figure out on this, where is the divergence between the Khoisan and everyone else? Well, probably somewhere in here. Okay, so this is where the Khoisan starts to change. Okay, somewhere in here, that's where you get your time from approximately. You can get your time in other ways from trees and that as well directly. Effective population size over here, I think it's double. That's in these units. It should be double that, 30,000. So that's the point.
So
That's the
100
split.
and... Oh, no, sorry.
The time units over here, I think that's, I think that might 200,000 or 300,000 years, depending on the calibration. That calibration seems to be highly dependent upon generation time, and generation time fluctuates within human lineages.
So that is that's kind of part of what's motivating his question. So, he's
So
curious
it looks like if we take, where was the divergence of the Denisovan and Neanderthal looks like it's here, which would tend to imply that the out of Africa with Heidelbergensis was possibly back here, possibly, although that's getting rather old. It looks like this is the long-term history of our lineage through time. And that's a sort of period before sapiens arises. And as I say, this is gross. There might have been a dip in there. Because at this resolution, we wouldn't necessarily pick it up. But even then, that's a fairly modest population size. If we really were drawing variation from all over Africa, or even all over southern Africa, you would think that the effective population size would be markedly bigger than that. Also, it doesn't explain why, if we originated in one part of Africa, why we got that sort of structure that we get, which almost looks like sequential migrations, and drops in effective population size along those internal branches as we hop through Africa and hop out of Africa. That needs to be explained as well. this is one way of estimating ancestral population sizes. There are others, and you can put them on a tree and you can associate them with edges on a tree. Okay, so a lot of work, a lot of methodological work to be done in this area. But the general picture seems to be, wow, it's just not that big.
So he also, and this is kind of part of the same question, he's curious what your data can tell us about kind of the emergence of the suite of life history traits that humans have that seem to have made them very good at expanding into a lot of different ecologies, etc.
Well,
right. So when did our interbirth interval go down? Cooperative care, etc.
Okay. Yeah. A lot of that is mysterious. Okay. Because all we know is that if we define Homo sapiens this way, we can talk about what we all had.
Right.
We can't talk very well about what we didn't have. Now, there are some studies now on Neanderthal skulls because we've got a reasonable collection. I mean, I'm still talking about 40 maybe skulls, maybe even less, but we've got some juveniles and we've got tooth eruption, which they are using to date. And I believe the consensus is it was kind of like Homo sapiens. What's also interesting is generation time. Okay? That's related to some of your questions, the generation time.
Right.
Well, what is interesting is if you try and calibrate these trees going back to chimp, then your dates look to be about a third more recent than they should be based upon trio sequencing. What is trio sequencing? That's when you take a mother and a father of a living person and you sequence the offspring and you figure out how many mutations per generation. And that's been done with populations all over the world now. And when you do that, you get numbers like this. If you do the split from chimpanzee, you tend to get numbers like this. The best explanation at the moment is that somewhere back here, the average generation time in our lineage changed from being something chimp-like, which might have been around 20 years, to something closer to a typical modern human around 30 years. And it seems to have happened before these because when we look at the branch nets going towards these, they all look about right. They're fluctuations, but they all look about right. There's no clear trend. Does that kind of help? That kind of suggests that somewhere back in here, we had a longer generation time which starts to suggest maybe slower development and longer lifespans, slower maturation perhaps.
That's also making the assumption, though, that the mutation rate per generation is constant.
Yeah, per generation is constant. Yeah, per generation being one of the critical things. A lot of the mutations come in. Now, that's a bit of a simplification. We know that as males get older, particularly, more mutations enter the germline. So if you've got a polygamous society and the average breeding age of the male is, say, 50 years, you're actually potentially going to increase your mutation rate on that lineage rather than decrease it. You'd think you'd decrease it because his generation times 50, who knows what the woman is, maybe 25 or something. But he's accumulating a lot of mutations.
Would that also be affected by, so I know we're talking about effective population size, but if you have a breeding population, so say we're talking about like tribes of people or things like that, where you have potentially much smaller sizes that are sort of inbreeding and things like that.
Yeah, structure inside, but the other one is also mating patterns. What is interesting is that through long periods of time, and we do actually see a few changes when I've reconstructed some of it. Actually, do see some changes in different parts of the tree. But mostly in this piece of the tree, the effective population size of males to females is 1 to 2. Okay? So
that means there's a high variability in males.
There's a much higher variability, basically twice the variability in a man's number of offspring to a woman's. Now, most of this history we think was hunter-gatherer. So pre-polygamous societies which tend to be more agricultural, settled. So hunter-gatherer societies, even in those, there does seem to be strong competition amongst men, and the ability to have more or less than the average number of children.
Or even higher mortality, right, among men? Would that also?
Yeah, basically competition fighting. Yeah, sometimes. Yeah.
Or even just dying through hunting. Or,
Yeah, it could be. Yeah, all of these factors tend to conspire against men. Okay, so men are the evolutionary suppressed sex. Biology discriminates against men, and yeah, so that seems to come through to some extent. That hopefully answers some of that question.
I think it does. Well, if Chris is still on there,
it's tough because there's a lag of about 5 minutes on here, so it's always hard to know if someone. It's like a second. We're playing a game of telephone here.
So what we should do is, I'll take any other question. We'll jump back to Chris as he's got a follow up. Any other?
Oh no, he says thanks. That was streaming live, so that was useful. So
okay,
I think we're good.
Cool.
So I'm still unclear. I might be misunderstanding something. So you're saying that the effective population size of our lineage is about the size of subspecies of chimpanzee, but sort of the general impression in our field is that we're so ecologically and reproductively successful, so why wouldn't that effective population size be a lot bigger than a genus subspecies?
Good point. Okay, it's all about history. If we originated from a population in Africa that was localized to, say, an area like Zimbabwe, and we didn't mix hardly at all with anybody else that was around, then all of our deep history comes from that lineage. Now, it also, because of these methods we talked about over here, you can trace that back. So this period right before we originated wasn't necessarily even the only bottleneck-like thing. It really does look like through this whole period, remember this is Heidelbergensis, like, we may be looking at sequential populations of 30,000 or less that evolve. Who knows how? Because we're not big populations. We don't evolve like bacteria. But we did evolve. And we seem to have evolved some pretty interesting traits. And not just Homo sapiens. Neanderthals are showing evidence of doing some pretty interesting things. And actually Neanderthals were looking kind of successful before they met us. They were spreading over Eurasia, possibly. So this seems to suggest that it's not like there was ever a Pan-African period in our lineage. That the whole history of Homo may have been, at some point we'll split off, and whatever genetic evolutionary process we're subject to, it may have worked kind of well in that sort of population size, right? Because it's kind of really hard to put things together across a big population if you need to select for multiple things. And so maybe a lot of the history of Homo may have been of kind of competing lineages. Now, the real question is, is this effective population size really suggesting a restricted area? Or is that what we should expect from, say, the whole of southern Africa 200,000 years ago, given climatic variation? Okay, I don't think the absolute answer that is in yet. What we do know is we're picking up other lineages, the Denisovan, Neanderthals, we're picking up fossil evidence of other things, we're picking up a little bit of genetic evidence coming in, that there were other discrete lineages out there. And maybe a lot of them, a lot more than we appreciated. And of all the mixings we know of, and there's now over half a dozen, including that one I mentioned between the Denisovan and an erectus-like thing, right? That's early. That's probably 400,000 years ago. And yet, it's only a few percent. So you can't just argue it's modern human culture that keeps us separate, right? So it looks like when Denisovans invaded the home territory of the lineages that were there before, they only picked up a few percent. Despite the fact that the other lineages had a severe hometown advantage, right? They'd been evolving there maybe for 1 million years or more. They were adapting to the local situation.
But so when you look, if you look at this graph and like, I'm still, I have some comprehension of what's going on, but you see this Denisovan Neanderthal drop off there. You see that at the same time as this expansion in the effective population size of these different sort of subpopulations of Homo sapien, right?
Well, no, these are the subpopulations of Homo sapiens through here. This is the common population probably up here.
Right, but like you really see, you see kind of a drop off. Basically what I'm getting at, kind of like the point I'm trying to make, is maybe you also see this expansion, or what looks like an expansion of the Homo sapien population size as you see a decrease in the Denisovans and that. So maybe they're just getting swarmed out. Like
No, completely different environments. Completely, almost no genetic interchange. We're talking about lineages that invade Eurasia. And actually, probably northern Eurasia. Well, northern Eurasia, during the ice ages and things, this suggestion from the archaeologists is that these were low effective populations, low impact on the environment. The number of Neanderthals, maybe 1/10 the number of Cro-Magnons. I don't know. The numbers are coming in at different times and long term. So it may just have been that these are northern Eurasian lineages, two very distinct ones that come from a Homo heidelbergensis-like ancestor. What we don't have is southern Eurasia, which potentially was a much better environment during the ice ages. We got archaeology, we see lots of hand axes and things in some places, but so far we don't know what they would have looked like. They may have actually looked like this, right? So whatever went into, say, Java or, that part of the world, if it was part of that invasion, maybe its population was like that, like a orangutan subspecies, right? So we know what the effective population size of orangutans is. It's quite reasonable. Well, what if a lineage from a heidelbergensis-like ancestor, maybe closer to Denisovan, got into Southeast Asia? What would be the effective population size? I don't know. I would think that it could exploit the environment maybe as well as an orangutan, particularly because the ice ages tended to open up more grassland, less forest. And the general impression is that these scattered savanna-like environments, as opposed to deep rainforest, is where Homo does well. That's why the question of when did the pygmies get established in the rainforest is such an interesting question. They're adapted to the rainforest, it seems. Their stature and other things suggest that there's been long-term selection for living in a forest, but we don't know. Their lineage is old and deep, possibly 160,000 years, but we don't know when they got into the forest. Because archaeologists often say, Homo doesn't do well in forests. Well, we may have to modify that at some point. Bones don't survive well in forests.
Right.
Stone tools are hard to find in forests. There's stuff growing everywhere. Rift valleys are great. Rift valleys, everything erodes out. It gets buried and it erodes out. Okay? So we also have some biases about where we're finding things in that. So Homo, all I'm saying is that the evidence is starting to suggest Homo was a pretty complicated, if you don't want to say specios, many independent lineages perhaps, doing a variety of things. The Floresiensis and the Naledi things, almost, you can't argue against those, but as we break it up and further in time, we're starting to see evidence that this may have been the norm. Effective population sizes like this, like a chimp subspecies, were the norm. But what's interesting is that they didn't recoalesce. So if you can imagine, will in the future, if we just disappeared and everything wasn't screwed up on the planet, would the chimps recoalesce? I don't know. Or maybe only one of them would survive. But I think Homo was a bit more adaptable. So I think when a lineage didn't survive in Homo, it may have had an awful lot to do with another Homo lineage moving into the area, not simply that the environment deteriorated. And that's certainly what we seem to see with Neanderthals and Denisovans, and maybe the West African example, and maybe Naledi as well. Okay, that our ancestors, or members of our species, were bad news to these other lineages. But we came from a very limited area.
So we're going to have to cut it off because there's another group coming into the room shortly. But thank you so much. That was really interesting. I think Chris signed off. So.
Yeah.